Hi guys, we are in week 14 now. We are making our way through the second 9 weeks. We're going to be looking at notes on complex inheritance today, and these are going to be under Wednesday's tab. And I ask you to please watch the video for complex inheritance. You're not going to need to write anything down, just make sure you pay attention. I also have these slides here for you as well, and you will need to use those slides next week whenever you answer your questions, your comprehension questions. So let's see if we get into this PowerPoint here. We've done simple inheritance. We know all about that now. So now we're going to talk about a little bit more complexity with things. So in this section, we're going to look at how one gene can affect the expression of a trait. Then we will look at how multiple genes can have the additive effects on a single trait. Sorry guys, I just wanted to make sure that I'm not supposed to be writing any dihybrid crosses for you or anything like that. And I luckily don't have any of that in this one. So nearly all traits are influenced by multiple genes. For example, height, production of growth hormones, cell division, uptake of nutrients, metabolism, etc. Just to figure out your height, all of those things have to take place. You have to have growth hormones, your cells have to divide, you have to have the right amount of nutrients, your metabolism, you have to be creating energy. If you don't have anything like that, then you're not going to grow taller. A defect in any of these genes would have an impact on an individual's height. So how did Mendel study height in a pea plant? He had tall pea plants and he had dwarf pea plants. So he looked at his true breeding strains. For a plant to be tall, for example, it would have all of these genes here were homozygous dominant, because it's true breeding, so that means it has to be homozygous, but all of these traits here affected the height. We're looking at a short plant. It has almost the exact same traits, except for at the very end here, it has a homozygous recessive trait for being a dwarf plant. So only a gene at one loci ended up changing. And you're going to end up mapping out genes here in a bit. So if you look at the locus for flower color, and locus is just an easy way to say location. The allele for purple flowers is going to be in the exact same place that the allele for white flowers will be. However, that gene is going to be different. Example, homozygous dominant versus homozygous recessive. Epistasis is a type of gene interaction in which alleles of one gene are going to mask the effects of a dominant allele of another gene. This is where you have two different genes are involved, and this is going to be an example in a dihybrid cross. Now I have that first bullet point there in a different color. That's just to draw your attention to it. I'm not making you take any notes on this section, but whenever you go to do your questions, those colored parts may really help you out. So if you look at this here, we have white variety number 1 mixed with white variety number 2. So we have 2 capital C's, 2 lowercase p's gave us a white flower. 2 lowercase c's, 2 capital P's also gave us a white flower, which is kind of crazy, right? Whenever we mixed those 2 flowers through self-fertilization, we ended up with a purple flower, which is a heterozygous type of flower. So if we look at what could potentially happen here, if we have all homozygous dominant alleles, that's going to give us purple. If we have any type of dominant allele, at least for one of the traits, it should end up giving us a purple flower as well. But if we have a mixture where we have 2 capitals and 2 lowercase ones, now we're going to get involved with the white flowers. And if they're all lowercase or all recessive, you should end up with a white flower. So you have to have 2 of them that are going to be lowercase letters homozygously. So we have 2 lowercase c's, that would give us a white flower, even though we have 2 capital P's. Again, here we have 2 lowercase c's, we have 1 capital, 1 lowercase, that still gave us a white flower because we have these 2 lowercase c's. Whenever we're looking at our purple flowers, you have to have at least 1 capital letter in each of the 2 traits. You have to have 1 capital C and 1 capital P in order to get that purple flower. 2 or more different proteins are involved in a single cellular function. They come together to form a single product, and when one doesn't work, the other cannot work either. Polygenic inheritance and environmental influence. So this is going to be talking about discontinuous traits, and these are phenotypes that do not overlap. They're not exactly clearly defined. So for example, our purple flowers or our white flowers. Although these are what you are most familiar with, most traits are not all purple or all white. It just, our genes don't really work like that. They teach you that stuff in biology, even though it's not 100% correct. It's just the easiest way for younger minds to comprehend bigger pictures. So we have something called a continuous trait, or you could call it a quantitative trait. And quantitative normally talks about numbers. So to show continuous variation over a range of phenotypes. For example, in humans, you could have height, weight, skin color, heart size, metabolic rate. An example in domestic animals would be the amount of milk a cow produces, numbers of eggs a chicken can lay, or the number of apples on a tree. Quantitative traits are polygenic. That means that there are several genes that are going to contribute to the outcome of that trait. It's not just one gene. Your eye color is actually polygenic. So genes contribute to the phenotype in an additive way. Environmental factors are going to play a huge role in polygenic traits. For example, the amount of sunlight or the amount of rain will affect how many apples a tree can produce. If you stick an apple tree that's supposed to give off a lot of apples every year and you put it in a desert, well, that tree is not going to do very well just based off of its environment, even though genetically speaking, it is engineered to produce. Skin pigmentation. This is going to vary substantially in any given population, and it's a really good example because you guys are familiar with so many different skin pigmentations. A simplified example would be a trait that's controlled by 3 genes, each of which have a light and a dark allele. So whenever we're looking at these traits here, we have trait A, trait B, and trait C. And then it's either going to have a D or an L. The D would be for dark, the L would be for light. So each one of these can have a different letter. You could have 2 Ds and an L, or 2 Ls and a D, or all 3 Ls or all 3 Ds. All these alleles are going to encode for enzymes that cause synthesis of skin pigment. Enzymes for dark alleles are going to cause more pigment than the enzymes for the lighter alleles. So if we look at that in a graph, we end up with a nice bell curve here. So you have your really lights, there's not going to be too many of those. You have your really darks, and there's not going to be a whole lot of those. And then gradually there's going to become more and more and more until you have like this nice mid section here where the majority of people would probably lie. Now this is the same bell curve, but with a sunnier environment. So depending, and I took this right out of a book of mine, but depending on whether or not, you live closer to the equator, you may have more exposure to sun. Naturally, your skin is going to adapt to become a little bit darker just to save itself from getting burned. So normally your bell curve would lie right here with this first one, where you would have like the medium skin tone being the most popular. But if you live in a place where there's a lot of sun, that bell curve is now going to shift. So you're hardly going to have no people that are completely white over here, hardly any people that are lighter skinned, and your medium range would actually be more on this darker side. And that's all based off of the environmental conditions. Violation of Mendel's law of independent assortment. So Thomas Hunt Morgan proposed the idea of linkage. And linkage is a tendency of genes that are close to each other on a chromosome to be inherited together. A linkage group is a group of genes that usually stay close or stay together during meiosis. So this is the formation of the egg and sperm cells. For a quick background here, we have Bateson and Punnett. Punnett was actually a guy's name. That's where we got Punnett squares. They had the first study showing linkage. They crossed peas observing color. You had your purple and your red, and they looked at pollen type. So they were looking at 2 different traits here, color and pollen type. A cross between a purple long and a red round flower produced all purple long flowers. When the F1 self-fertilized, So they would have been heterozygous for these traits. They would have been hybrids. They found that PL uppercase letters and PL lowercase flowers occurred more frequently than P capital P lowercase l or lowercase p capital L, which sounds kind of strange because if we look at independent assortment, that says that everything is going to independently go into the cells on their own, that they're not actually linked to anything. Each trait is its own entity. So they're deciding, okay, well, that's not actually true. If you have two dominant traits, they're more likely to stick together. And if you have two lowercase traits or two recessive traits, they're more likely to stick together. But mixing the two traits really doesn't happen very often. So here is what they crossed here. We have the purple long and we have the red round. When they crossed it, they got all purple flowers that had long pollen grains, the heterozygous form. They allowed them to self-fertilize. And you ended up with more purple flowers that had long pollen grains. Whenever you allowed them to self-fertilize, you ended up with a lot of purple long flowers, not too many purple round, not too many red round, and more red round flowers. Did I say red round? Not too. So we didn't have too many of the heterozygous forms here, and these are our two heterozygous forms. The recessive traits kind of stuck together, and the dominant traits tended to stick together more often. If you couldn't see that other example too well, here it is written out a little bit better. These are the true breeding lines. This is what they crossed the first time. Whenever they did their F1, this is what they ended up with. And then they allowed that to self-pollinate itself. And your expected ratio, when we do ratios, it should be a 9-3 to 3-1, but that wasn't really exactly what they got. They observed that there were definitely a lot of the homozygous dominant forms of the trait. There was more homozygous recessive than there was heterozygous traits. So they're deciding, okay, maybe these things don't independently assort. Maybe they are linked together. So we're going to move away from Mendel for a little bit here, and we're talking about linkage according to Thomas Hunt Morgan. He studied linkage in flies, similar to the results of Bateson and Punnett, and he proposed 3 ideas. He said different traits located on the same chromosome are more likely to be inherited together. Crossing over is going to allow for new combinations of alleles, and the likelihood of crossing over depends on the distance between the two genes. So he did this experiment with these flies. He had a wild type of fly, and he had this black vestigial type of fly. And you can see where the genes are located at on each chromosome. The parental genotypes would have been these two here. Here's your wild type, here is your black vestigial. If we did a recombinant genotype where we kind of pulled them apart and mixed them together, and you can see it really well, the two different colors of the chromosome here, it shows you one's coming from mom, one's coming from dad, and this is the example of crossing over. So whenever you do that cross, you should end up with a 9-3 to ratio. But they ended up with black vestigial flies, and they ended up with the wild type of flies being the most prevalent, most dominant. And your recombinant types, there were not too many of those. So the heterozygous forms were not as prevalent as they should have been. They should have been overruling on this black vestigial one. There should have been more of these two in comparison to this one here. So Morgan's test on flies, body color was gray versus black, wing type was straight versus curved. Baby flies that have the same traits as the parental generations are known as non-recombinant. Baby flies that have a different combination of traits are recombinants, or they are non-parental types. So they're the hybrids. They're a mixture of both the mom and the dad fly versus being all mom or all dad. Gene linkage mapping is an experimental method used to determine the linear order of genes that are linked to each other on the same chromosome. Recombination frequency is the frequency of crossing over between two genes. You would use a test cross to estimate the distance between these two genes. You will have to do some math like this later on whenever you're looking at map distance. So the number of recombinant offspring divided by the total number of offspring times 100. So for our map distance, we're looking at recombinant offspring. So now these are recombinant phenotypes up here. So I take 206 plus 185, and I'm going to divide that by the total number of offspring. So then I would add up all three of my offspring types on the bottom, divide those two, multiply that by 100, now I get 17.0 map units. One map unit is equivalent to a 1% recombination frequency. So that means that there's a 17% chance that you're going to have a hybrid form. The odds are not too good to have these recombinant phenotypes. The purpose of doing that is to analyze genes of an organism that are easily crossed and produce many offspring in a short period of time. So the next law that we have to talk about we're in violation of is the law of segregation. So we're looking at extranuclear inheritance. This is the transmission of genes located outside the cell nucleus. And that's going to sound really confusing to you because up until this point, all we've talked about is how DNA is only located in the nucleus. The nucleus is what goes through and copies the DNA, and that's what divides it. And then you end up with, your genes coming from mom and dad. So extranuclear inheritance, getting back to that, is the same thing as cytoplasmic inheritance. So this is where you have genes that are outside of the cell's nucleus. Each organelle kind of has its own little genome here, and they are much smaller than the nuclear genomes that we have in our nuclei. It contains information vital to the function of the cell. For example, the two main ones that we talk about are the chloroplast and the mitochondria. Both of these have their own sets of genes, and it's inherited. Most of the time they call it motherly mitochondrial DNA, because you do get your DNA from your mitochondria from your mom. Dads don't get to give you any of that stuff. I'm not exactly sure if it works that way in plants, but you get the general idea here. Carl Correns observed leaf pigmentation in the 4 o'clock flower plants, which is a pattern of inheritance that does not follow Mendel's law of segregation. The pigmentation for the offspring depended solely on the maternal parent, which is maternal inheritance. So here are those 4 o'clock flowers that he was looking at. And you could have either some white leaves or you could have green seed leaves. So he noticed that whenever he was crossing things, here's the dad, this is going to be the mother. Whenever you crossed these two plants here, you ended up with white. Whenever you crossed a green dad with a white mom, you still ended up with a white child. Whenever you crossed a green mom with a white dad, you ended up with a green child. And when you crossed a green dad and a green mom, you ended up with a green child. So whatever the mother was is what those children ended up having. The dad's genes did not actually play a role in that at all. The majority of plants do exhibit maternal inheritance for the zygote's cytoplasm. Biparental inheritance is when both pollen and egg contribute chloroplast to the offspring. Paternal inheritance only goes along with the pollen because that is the male portion of the flower, so the dad plant gets to control the pollen. We're almost done here. And I know this is a lot to take in at one time. Don't worry, gene mapping and gene linkage when we actually do examples are not this confusing. So epigenetic inheritance is a modification of a gene or chromosome during egg formation. Sperm formation or early stages of embryo growth alters gene expression in a way that is fixed during an individual's lifetime. It is permanent for the individual, but it is not permanent for their children or for their children's children. It's only permanent for that person. X inactivation is one X chromosome in the somatic cells of female mammals is inactivated or not expressed. Somatic cells are like regular skin cells. A bar body is a structure found on the cells of female cats, but not male cats. And calico cats are going to show patchy colored fur due to the 1X not working properly. So we talk about calico cats a lot when we look at X inactivation because those X's are actually going to play a role in how the cat is pigmented. For female mammals that are heterozygous for X-linked genes, they have their somatic cells that will express one allele, while the other half will express the other allele, which is why you end up with that patchy coloring on those cats. Mosaics are heterozygotes composed of two types of cells. You want to think of a mosaic artwork. So here we have twin, yes, that's right, they are twin baby kitties. They are female kitties, and they look nothing really alike other than the fact that they have the same colors in them. But it's all about the X and activation. So any place where the X is not working properly, they're actually white. Black and the orange color are going to be codominant traits. X-linked disease with X inactivation. It's possible for a female to express a recessive X-linked disease even if they are heterozygous for the trait. That happens if they have X inactivation. So if their 1X isn't working properly, then they're going to show that recessive trait. Researchers believe X inactivation occurs because of a dosage compensation. This is because X chromosome is very large in comparison to the Y. If you inactivate one of these X's, it's going to reduce the number of the expressed copies, which is the dosage. So you're going to decrease the dosage from 2 genes down to 1 gene. And it makes X-linked genes in females and males more equal. Not that we necessarily want to be more equal to a guy when it comes to our genotypes, but I guess it's easier for our bodies to express that. In humans, the mosaicism is usually advantageous, protecting carriers from the traits, although X inactivation can occur. And it's very rare. Normally, it's seen in renal diseases and in different skin conditions if you have this X inactivation. Genomic imprinting is a situation in which a segment of DNA is imprinted or marked in a way that affects gene expression throughout the life of the individual who inherits the DNA. It can involve a single gene, part of a chromosome, a whole chromosome, or all the chromosomes inherited from a single parent. It's permanent in the individual, but it can change from one generation to the next, and it does not follow the Mendelian pattern of inheritance. In fact, nothing with complex inheritance is going to follow Gregor Mendel, who we idolize for being the father of genetics. So an example of imprinting, and this looks awfully confusing, and I'm just going to read through this, and you guys can read through it on your own as well. In mammals, the growth factor LGF2 interacts with the LGF2 receptor. Makes sense. You have an enzyme or something, and it comes in contact with its receptor, and they link up. Deleting the mother's LGF2 receptor genes produces overly large offspring. Deleting the father's LGF2 gene produces dwarf offspring. Deleting the mother's LGF2 receptor gene and the father's LGF2 gene produces normal-sized offspring. So you need both cases in this particular example. The imprints on the LGF2 and LGF2 receptor genes normally cancel each other out. Changing the imprint on one copy of the gene has a dramatic effect on the size of the offspring. And this result supports the genetic conflict hypothesis. So you need the mom's receptor to not be working and the father's gene to not be working, and then you end up with a normal baby. So whenever we have the dad up here at the top, paternally expressed, and the mom is over here on the side. So whenever both of them are present, you get a normal baby. When the mom's is present and the dad's is not, you get an absent, or dwarf baby, sorry. When the dad's is present and the mom's is absent, you get a large baby. And when they're both absent, you get a normal baby. So you either need them both to be there or both not to be there, otherwise your babies are going to be weirdly sized. Maternal effect genes, this is the inheritance pattern in which the genotype of the mother determines the phenotype of the offspring. Maternal cells, called nurse cells, surround a developing egg and provide it with nutrients. The nurse cells give their genes to the egg during embryonic development. Gene products of nurse cells determined by the mother's genotype influence the development of the offspring. Everything is based solely off of the mother's phenotype here. So we have a mother dextral cell and a father sinistral cell. When you mix them together, they end up heterozygous, looking like the mother and the father. And whenever you allow them to self-pollinate or self-fertilize, not self-pollinate, we're not talking about plants here, these are shells, you end up with everybody looking like mom, but this person over here has a genotype of dad. However, phenotypically speaking, they look like mom. Mothers have a lot of control based off of those nurse cells. So they're not really sure what happened there. So that is it for our notes on complex inheritance. And I'm sure that all sounded very confusing to you and you probably don't understand a whole lot of it. That's okay for right now. We're going to look at some gene mapping tomorrow. We'll do linkage next Monday, and then we're going to move on and do some Google questions. And that'll be the end of our section on complex inheritance.